This is Agriculture Today, and I'm Shelby Varner with the K-State Radio Network. Starting today's show is K-State canola breeder Mike Stamm to talk about the current state of the canola crop in Kansas. He says what the crop has experienced and what growers need to keep an eye on moving forward. Information about a newly developed Bovine Respiratory Disease Vaccine from Louisiana State University professor of molecular virology Shafiqul Chowdhury keeps the show going as he talks about the benefits. Now is the time to take steps to control fly breeding on dairy farms. So to end today's show, K-State dairy specialist Mike Brouk recommends focusing on three things: where flies rest, feed, and breed. That and more is coming up ahead. You're tuned in to Agriculture Today, and we continue our show talking about the current state of the canola crop. And then to talk about it, we're joined by K-State canola breeder Mike Stamm. Mike, thanks for joining us today.
Glad to be here.
Mike, how is the canola crop looking right now?
Right now it's looking bright and yellow, which is the most photogenic period, of course, for the crop, which is a good thing. It's a really critical time, though, for canola, with it being peak water demand. This is the time when we need moisture available because we're in that reproductive phase. And so there could be any number of stresses that cause yield loss at this time period. So it's a really, really important time to get this crop to the finish line.
And is the cold weather we had a few weeks ago something to be concerned about?
It is. It always is with our winter crops when we have these spring freeze events in March and April. I think this year, though, we're pretty fortunate with where the canola crop was at. The damage depends on, the ultimate low temperature, the duration of that temperature, and then the growth stage of the crop. And this year, the crop was not quite as advanced as we've seen it in some of the previous years where we've had some of these bitterly cold temperatures. Now, it did get downright cold in central Kansas. We measured 14 degrees at Hutchinson on the 14th. And so I was certainly concerned. But when I evaluated that field, the crop was in that early bolting to early flowering stage. And, at that time period, the crop is actually fairly tolerant to these hard freezes.
And what is that threshold for their tolerance?
We start talking about spring freezes. If we get down into the mid-20s, canola will rebound from those freezes with relative ease if conditions become favorable after the event. If we start talking about teens and lower teens, then we've got some real concern. And because we've seen that in the past where we were just bitterly cold and maybe at a more advanced growth stage than what we are this year, maybe in the flowering stage, you put the bitter cold and that late flowering stage together, and you can see some real negative impacts. But fortunately, even though we had that low temperature this year, I think that being at the growth stage we were at kind of saved us. Are
growers able to go out and check their fields?
Yes, and they should do that. And, with canola, the response, or actually the symptoms are a little bit more immediate than what they may be for wheat because you can see it. With wheat, you have to split stems and oftentimes to see the real damage from the freeze. But with canola, it's pretty visible. So, the first thing that I look at is the leaves, obviously. And I talk about in that e-update article I wrote about leaf discoloration or leaf bleaching. It's a loss of chlorophyll in the leaf. And so the leaves will look white, brown, and a lot of times you'll see within the center of the leaf that bleaching discoloration, but then also the tips are burned back. And at the stage we were at, the canola is still actually producing leaf area, and so it's able to grow through that bleaching pretty easily. And so it doesn't take long after the freeze event where you start to see more leaf area and the plants overcoming that effect of the freeze. Probably my biggest concern with this freeze event is the stem cracking. And when you evaluate the plants, you'll see on the main stem, oftentimes you'll see just a crack in that stem. and it's just from that freezing and thawing impact of the freeze. And so if that stem cracks, it could open up an opportunity for fungi to enter and cause stem rot, or it could weaken that stem. And then as the crop is adding weight in terms of pods and seeds, it might cause those plants to lodge then. I didn't see any stem splitting where the stem is completely split open, and that's the worst impact from these freeze events, when those stems basically fill with ice and that causes them to split wide open. That's a vector for stem rot and for a weakening of the stem and lodging as the crop advances. But I really didn't see any of it this time around, and I think, again, it was because of the growth stage we were at.
So should growers go out into their field and see if they possibly have a chance for fungal decay so they can plan ahead when it comes to harvest time?
It's just good to know. There's nothing you can really do to reverse the impact of that cracking or splitting open of the stems, unfortunately. But it's good to know if you see that in your field. And also at this flowering stage, you could potentially see if that impact on the stem was significant enough. You could see those plants even start to lodge initially. Like oftentimes we'll see the plants kink over, and then as conditions improve, they actually start to grow vertically again. And so you get this weird kinking of the stem, but the plant actually isn't dead. If you see that, when you walk up to a field, you'll know that there could be some impacts on those stems. But canola's a great crop in that it's indeterminate, and so it's got a lot of growing points where it can overcome a lot of these effects.
So overall right now, Mike, not a big impact from what you're seeing from this freeze we had towards the end of March?
I really don't see a major impact from it. Now, we will probably see minor impacts in terms of yield because as you move further south in the growing area, into southern Kansas, on into Oklahoma, the crop is further along, but they didn't get as cold. And even if we have a small or minor freeze event, I should say, you'll see those flowering racemes where you'll have a little bend in them that's indicative of cold temperatures. But then you'll also see at that bend, you'll see bud or flower or pod abortion. So there'll be a blank in that stem, but it'll be just a short area. And then above that, you will see normal bud or flower or pod development. And so, yeah, there'll be some loss of pods and then maybe a loss of yield as a result. But again, the crop has this compensatory ability about it, which we like for the southern plains because we have a lot of stresses, and we've seen that canola can overcome these repeated stresses. Anything our canola growers need to be thinking about as we move forward now? Well, we definitely want to be out there scouting for the possibility of sclerotinia or white mold, which could be a disease that impacts the crop as we move further into flowering. So that is something to be aware of, although we haven't had rain events that could potentially lead to a sclerotinia being a problem. So that's of lesser concern to me. The big impact right now would be insect pressure on the buds and the flowers and the pods as they are developing. So diamondback moth would be one that I would be concerned with. Also, lygus bug can be an issue. False chinch bug, since we're kind of on the dry side right now, they can become an issue. So those are the things that I would be on the lookout for. The biggest issue that I see moving forward right now is some timely rainfall. We really need that timely rainfall right now, like I said, to kind of get us to the finish line because we are at that peak water demand. So overall, right now, the canola crop's looking okay, just really hopeful for some water. I'm hopeful. Canola roots go deep. They really mine deeply for moisture, and so it's hanging on for sure, but a timely rain would certainly help. Mike, I appreciate you taking the time to join us today and share with us some information about the current state of the canola crop. Happy to be here.
That was K-State canola breeder Mike Stamm. If you'd like to read more about this, you can do so in an article from the April 5th Agronomy E-Update titled Spring Freeze Effects on Winter Canola. You can find it by going to eupdate.agronomy.ksu.edu, or we'll also link it in today's show notes on agtoday.net. Before we cut to a break, we're now going to be joined by Kansas Livestock Association's Scarlett Maydinger as she shares how professional sport dietitians are recommending beef for athletes.
Proper nutrition before, during, and after physical activity plays a crucial role in muscle, skeletal, and athletic development. To help share this message and educate active families on how beef can fit effortlessly into the diets of their young athletes, the Kansas Beef Council collaborated with 2 Kansas City sports nutrition experts. Leslie Bonsi, a dietitian working with the Kansas City Chiefs, wrote an article that covers the 4 P's of protein to show how high-quality proteins can influence post-workout recovery and help promote performance. She also shared her favorite beef recipes for athletes. Since recovery is such an important aspect of athletic training, Sporting KC dietitian Kim Gaskell wrote an article that took a deeper dive into how protein helps repair damaged tissue and build new muscle to ensure athletes are adequately prepared for their next training or game. Gaskell also noted in her article that beef burgers are a favorite among Sporting KC players. Both articles are being promoted through display ads on various websites, the Kansas Beef Council social media channels, and through a handout that will be distributed during coaching and youth athletic events throughout the spring. They also can be found on kansasbeef.org under Health and Wellness. I'm Scarlett Maydinger.
We're cutting to a short break now on Agriculture Today, but stick around because when we come back, we'll be talking about a newly developed bovine respiratory disease vaccine. You're tuned in to Agriculture Today, and continuing our show, we're discussing a recently developed bovine respiratory disease vaccine. And then to talk about it, we're joined by a professor of molecular virology at Louisiana State University, Shafiqul Chowdhury. Shafiqul, thanks for taking the time to join us today and share with us some information about this vaccine.
Thanks for the invitation and the opportunity. So just let me give you what's about bovine respiratory disease. So bovine respiratory disease is a complex caused by several viruses and ultimately a secondary bacterial infection that combined called bovine respiratory disease complex. And so among the viruses, there are 3 or 4 viruses. One of them is bovine herpes virus 1, also known as infectious bovine rhinotracheitis. And then there is bovine viral diarrhea virus, bovine respiratory syncytial virus, and there is also bovine coronavirus and bovine parainfluenza 3. So these are all viruses that can play a role. But the most important virus is first the bovine herpes virus. This is the herpes virus that when infects a host causes respiratory infection. And if it's a cow which is pregnant, then it can cause abortion. So about 10 to 12% animals will abort if they're infected. But this virus, the problem is the virus, when it infects a host, it goes in the nose, replicates there, causes some lesions, and then the virus travels along the nerve that innervates, nose area, and goes and hides into very close to the brain, in the base of the brain, where these neurons that connect to these fibers resides. And in that neuron, the virus lives then forever. You can never get rid of that. And human has that equivalent virus called herpes simplex, and that's why you get those cold sores. Now what happens is when you're stressed, then those viruses wakes up in that neuron and travels back to the nose and replicates. And then again shed through the nose, and that becomes a source of infection to the other viruses. So this is basically the field wild virus. And bovine viral diarrhea virus is also a respiratory virus. It causes respiratory infection, but one of the problems of that virus is that it also immunosuppresses. That means it will depress your immune system such by killing those cells that actually protects you. That invites then other infections because the protection level is low. Then bovine respiratory syncytial virus is another RNA virus. So RNA virus means it's a ribonucleic acid. This is DNA. Herpes is a DNA virus. So the RNA virus is also known to change a lot. And there's a reason why we don't want to talk about that technique, but they change a lot. So that becomes problem in the field because the virus, if it is changing, mixing, it becomes a problem to control that infection.
And what is the problem with that?
The problem with this virus infection is that when the animals get infected with any one of these virus, and more important is the herpes, they immunosuppress, and also herpes time to time come back, immunosuppress again, and those cattle can get infected with the secondary bacterial infection, which is called Mannheimia hemolytica. Now, that bacteria normally lives in the animal respiratory tract. However, when there is immunosuppression or some viral infection, they colonize or they go to the lung and produce a lot of bacteria and ultimately get pneumonia and they die. So that's the major problem in the field when the animals get infected.
And what is currently being done in terms of a vaccine?
The vaccine companies have their vaccine and they make it multivalent. In other words, they have all three viruses that causes the disease, or four: bovine herpes, bovine viral diarrhea virus. There are two of them, type 1 and 2, and then bovine respiratory syncytial virus. And then they also mix the bacteria, which is Mannheimia hemolytica, that ultimately kills the animal. The problem with those vaccines is that the way the vaccines are developed, they basically take the original virus that they isolated from the field, and they basically passage them in the cell culture many times, hundreds of times, so that they get adapted to the cell culture and loses their ability to cause disease. So when they mix those viruses together and inject into the animal, it basically induces immune response, which ultimately protects the animal from their infection. Initially, that sounds good, but what happens is that these viruses can also replicate and persist in the animals. For example, herpes modified live vaccine can go latent or reside in that neuron that I talked about and come back and time to time shed. And the BVDV and bovine respiratory, they're all immunosuppressants or other complications. And that the vaccine itself invites some other infections. And the mere fact is that when the vaccine virus is persisting and shedding and changing, especially RNA viruses, then the vaccines they develop currently in the lab, they won't work because the virus has already changed. So that's one problem. The other is sometimes the vaccinated element comes down to the disease even 3 weeks after. And we had a cattleman who has a farm in Texas call me and told me that he vaccinated recently, and 3 weeks later they came down. So this is a 50-plus years technology that is still being used by the vaccine companies, and cattle farmers are still using it because there are no other good vaccines available.
Shafiqul, what have you done then in terms of a new bovine respiratory disease vaccine?
My lab basically studied the pathogenesis, how the genes and what causes disease, and we did a 15-plus years of research before we started developing the vaccine. So what we did is that we changed the herpes virus, bovine herpes virus type 1, but we took away 3 or 4 genes we call quadruple mutants because we changed 4 genes or deleted completely or partially. That takes away the ability of the virus to immunosuppress or, viruses has to evade the immune system. So it takes the ability, and also it takes the ability of that virus to cause disease because some of the genes are important for pathogenesis. And most importantly, we also took away the ability for the virus to transport within the neuron from that neuron cell where the virus hides forever to come back out. Now, we cannot make a virus that doesn't go to that neuron, but we can make a virus that can never come out. So, in other words, this virus is just vaccinated, infects the animal, produces immune response, but cannot ever come out, which the other does. And also, this vaccine has a marker which allows it to be distinguishable in the field from the real virus, because herpes virus in Europe, for example, they eradicated this virus because they killed all the animals that has the real virus. and then they have a test that distinguishes whether it is a real virus or vaccine. So they can tell whether this animal is infected. Once they see that animal is infected, they kill it. If they're vaccinated, then they don't. So our vaccine virus has that. Now, to make a multivalent vaccine, what we did, we used this virus, which we call a virus vector, quadruple virus mutant vector, bovine herpes. We use this to deliver the proteins of other 3 or 4 viruses that I talked about in the form that those viruses will also produce if they infect cells. But these proteins that we put, they basically will mount immune response and prevent the infection of those viruses. So we eliminate the chances of those viruses that usually change, and 2-3 years later, your vaccine won't work because the virus has changed. And basically, this mixing in the field and causing more problems, that doesn't happen. But it will protect against those viral diseases. Now, if the virus in the field that this RNA virus has changed, we can also change our proteins. We can find that what kind of protein they're expressing now change. We can put that in. So that allows the ability to do that. And in the future, we also are planning to put the bacterial antigen, and this is in the process, in the pipeline. So what we are going to ultimately accomplish is we will have a vaccine in which we will have put antigens of those viruses that causes BRD or bovine respiratory disease and bacteria, and have a multivalent vaccine just like the companies are doing, but it will be safe. It will not cause problem in the long run, which happens with the other viruses. They change, they mutate, and cause problem because they also immunosuppress, which won't happen with this vaccine. So this virus is being more developed now, and the press release was actually on a technology that was 6 years ago. We have now come a long way, and we have even improved our technology where we can put 6-7 antigens from some other viruses in one particular virus like bovine herpes. So ultimately, we would hope to have this vaccine replace this problematic vaccine, which is a 50-plus years technology.
What is the future for this vaccine that you have developed, Shafiqul?
This vaccine is now in the process of licensing. There is a company who licensed this vaccine. There are some other companies, they are also interested, and the process is they're looking at the efficacy and safety, whether it causes abortion itself, because one of the things that vaccine companies would like to determine is that the vaccine virus itself may cause abortion. And we tested that by injecting pregnant animals. In our hand, it didn't, but they want to make sure that it does not kill even the embryo, that very early age of embryo development. So once these tests are done, they are going to apply for USDA license. And once that happens, then it could be commercialized and used in the field. So that is another important consideration here to think about how we can improve the vaccine and prevents these losses.
That was Professor of Molecular Virology at Louisiana State University, Shafiqul Chowdhury. I will put a link in today's show notes if you'd like to learn more about the work Shafiqul has done. We're cutting to a short break now on Agriculture Today, but stick around because we have more for you ahead.
This is Agriculture Today. Along with Shelby Varner, I'm Jeff Wickman. We end today's program with this week's milk lines. There are a number of steps that can be taken to prevent fly breeding on dairy farms. K-State dairy specialist Mike Brouk suggests focusing on 3 things: where flies rest, feed, and breed.
Today I'd like to visit with our Kansas dairy farmers concerning their fly control programs on their dairies for the summer. You know, there's lots of different products out there on the market that we can use in our fly control program on our dairy. We have baits, we have traps. Within traps, there are fly strips. There's sprays that we can use on our premise directly on the animal. There's dusts that contain chemicals that we can use on back rubbers. And we have ear tags that we can use. We can use some biological control, and then there's also larvicides we can include in the feed. And there's probably several other things we could maybe list on that list of many choices that we have. But here's what I want you to focus on. When you look at the fly control program on your dairy farm, you really need to look at manure management, fly prevention, and fly elimination. And it's really important that you focus on where do the flies rest on your facility, where do they feed, and where do they breed. So as we combine these areas and we consider what we really need to focus on, let's make sure that our fly control program actually addresses those key three areas: where the flies rest, where they feed, and where they breed. When we think about where they rest, we would like them to pick up some sort of chemical that might eliminate them. So this is where premise sprays really come into play and are very effective. So if we have a way to spray our facilities, the inside where flies might rest, that gives us the ability to transfer some chemical to those flies and eliminate those flies. Also, keep in mind that some of these products may discolor painted surfaces, so test a small area of the surface with the product that you intend to use to make sure that you do not have an issue there. Any light colors, generally white, will attract flies, so any areas that you have where you have this type of coloring might be important to pay attention to whether or not flies are resting there. So as we move on, we might think about areas where the flies will actually be feeding, and that includes your animals. Some of our flies, such as stable flies, tend to feed on the cow's legs and bellies, and these are biting flies and cause us lots of issues when we think of production. As you look at your dairy cows today, you may notice that they are starting to bunch in the pens. They'll be stamping their feet and their tails will be very active as they try to protect themselves from these biting flies. Other places that flies tend to feed are also on the face of the animal. So when we think of horn flies and we think of face flies, and even house flies, they will tend to feed around the eyes and the nose of the animal. This can cause pain for the animals if they're biting flies like horn flies, and it will result in losses of production. So in our dairy facilities, usually the house flies and the stable flies are the ones that are most likely to cause us issues and result in production losses. If we're dealing with pastured cattle, then the face flies and the horn flies would be a fly that we need to control. Finally, we need to think about how we're going to control flies where they breed. So when we think about breeding flies, that's going to be associated with spoiled feed and manure in general. So around our dairy, trying to eliminate those things as much as possible would be the first thing that we need to do. If we're trying to actually control the breeding of flies, then larvicides that we feed may actually help do that. One of the critical areas on your dairy you really need to focus on is the calf area. This tends to be an area that's often ignored with fly control that we do in the rest of the barns, but very important in trying to control the overall fly population on your dairy. So as we think about fly control on our dairies, again, I encourage our dairy farmers across the state to focus on manure management, fly prevention, and fly elimination, and making sure that you're considering what you're doing in each of these areas, where the flies rest, where they feed, and where they breed. This is Mike Brouk with K-State Research and Extension.
Thanks, Mike. And that'll do it for this edition of Agriculture Today. For Shelby Varner, I'm Jeff Wickman. This is the K-State Radio Network.